For over 70 years, doctors like myself have prescribed a medication called Hydralazine. It is a reliable and common drug used to treat high blood pressure. We have seen its effectiveness firsthand, especially in critical situations like preeclampsia, a dangerous condition where blood pressure spikes during pregnancy. The drug works as a vasodilator, meaning it helps to relax and widen constricted blood vessels. This simple action allows blood to flow more easily throughout the body, which in turn brings down a person's blood pressure. For decades, we knew what it did, but the precise molecular details of how it accomplished this feat remained a perplexing medical mystery. It was a tool we used effectively, but its inner workings were not fully understood. The long-standing puzzle has finally been solved by a dedicated team of researchers at the University of Pennsylvania. In a groundbreaking study published just this month, in November 2025, they pinpointed the exact mechanism of action. Their work revealed that hydralazine functions by blocking a specific enzyme in the body, known as 2-aminoethanethiol dioxygenase, or ADO. This enzyme plays a key role in the process that leads to blood vessel constriction. By inhibiting ADO, hydralazine essentially cuts off the signal that tells the blood vessels to tighten up. This discovery is a significant moment in pharmacology, finally providing a clear scientific explanation for a drug that has been a staple in medicine for generations. Understanding this mechanism is more than just solving an old scientific riddle. It opens up entirely new avenues for how we might use this familiar medication. The researchers' discovery that hydralazine targets the ADO enzyme has profound implications. It confirms why the drug is so effective for hypertension, but also shines a light on other biological processes where ADO is active. This new knowledge provides a foundation for exploring hydralazine's potential beyond its traditional role in managing blood pressure. It is a classic example of how revisiting old medicines with modern scientific tools can lead to unexpected and exciting breakthroughs that could change patient care in the future. The journey to this discovery involved meticulous laboratory work and a deep dive into the drug's molecular interactions. Scientists used advanced techniques to observe how hydralazine binds to and deactivates the ADO enzyme. This finding is not just an academic footnote. It provides a roadmap for developing even better drugs in the future. Now that we know the target, it may be possible to design new medications that are even more selective and potent in their ability to block ADO, potentially with fewer side effects. This elegant piece of science, as one expert described it, has peeled back the curtain on a 70-year-old mystery, setting the stage for the next chapter in hydralazine's long and useful history. The discovery of hydralazine's true mechanism did more than just explain its effect on blood pressure. It unveiled a completely unexpected link to one of the most challenging diseases in medicine, glioblastoma. This is a highly aggressive and fast-growing form of brain cancer. For patients diagnosed with glioblastoma, the prognosis is often grim, with a median survival time of only about 12 to 18 months. The 5-year survival rate is tragically low, hovering around just 5%. This cancer is notoriously difficult to treat because its cells grow and spread rapidly within the brain, making complete surgical removal nearly impossible and often resisting traditional therapies like chemotherapy and radiation. The crucial connection lies within the ADO enzyme that hydralazine blocks. The research team found that glioblastoma tumors often exhibit very high levels of ADO activity. This enzyme appears to help the cancer cells survive and thrive, particularly in the low-oxygen environments that are common inside a rapidly growing tumor. By producing this enzyme in large quantities, the cancer cells essentially create a protective shield that allows them to adapt and continue their relentless growth. This insight into the tumor's biology revealed a potential vulnerability, a target that no one had identified before. It suggested that if the ADO enzyme is critical for the tumor's survival, then a drug that inhibits it could be a powerful new weapon. This finding immediately raised an exciting possibility. Could hydralazine, the old blood pressure drug, be repurposed to fight this deadly brain cancer? The logic was straightforward. Since hydralazine is a known inhibitor of the ADO enzyme, it could theoretically disrupt the very mechanism that glioblastoma cells use to stay alive and multiply. This is a prime example of drug repurposing, where a medication approved for one condition is found to be effective for another, completely different disease. This approach is incredibly valuable because it can significantly shorten the time it takes to get a new treatment to patients, as the repurposed drug often has a long history of safe use in humans. the idea of using a simple, inexpensive blood pressure medication against a complex cancer like glioblastoma is a game changer. Current treatments can be incredibly expensive and come with severe side effects. Hydralazine, on the other hand, is globally available, affordable, and has decades of safety data behind it. Dr. Walidin Sivakumar, a neurosurgeon who is not involved in the study, praised the research for uncovering what he called a completely new vulnerability in aggressive brain tumors. The potential to leverage an existing, well-understood drug gives researchers and clinicians a significant head start in the battle against this devastating form of cancer. The most fascinating aspect of this potential new treatment is how hydralazine appears to work against glioblastoma cells. Instead of killing the cancer cells directly, which is the goal of most traditional chemotherapies, the drug does something entirely different. When the researchers treated glioblastoma cells with hydralazine in their laboratory studies, they observed that the cells entered a state known as senescence. In simple terms, the drug caused the cancer cells to go to sleep. These senescent cells are still alive, but they stop dividing and become dormant. This effectively pauses the tumor's growth, preventing it from spreading further. This concept of inducing dormancy represents a major shift in cancer treatment strategy. The conventional approach has always been to try to eradicate every last cancer cell, a difficult task that often causes significant damage to healthy cells in the process. The senescence-based approach, however, aims to manage the cancer by putting it into a permanent state of hibernation. In the lab, the researchers found that a single dose of hydralazine was enough to keep the glioblastoma cells in this dormant state for several days. This suggests a potential treatment that could transform glioblastoma from a rapidly lethal disease into a more chronic, manageable condition, much like we manage other long-term illnesses. While this strategy is promising, experts also urge a degree of caution. Dr. Nicholas Kleiber, a physician who reviewed the study, described the science as rigorous, but pointed out potential challenges. Inducing senescence to control a tumor might require continuous therapy to keep the cells dormant. If the treatment is stopped, there is a risk that the cancer cells could wake up and begin dividing again. Furthermore, glioblastoma is known for its high mutation rate, meaning it can quickly evolve and develop resistance to drugs. It is possible that some cancer cells could find a way to bypass the effects of hydralazine and resume their aggressive growth, requiring new strategies to keep them in check. Despite these challenges, the potential benefits of this approach are enormous. By not trying to kill the cells with toxic chemicals, a senescence-inducing therapy could have far fewer side effects than traditional chemotherapy. This would significantly improve the quality of life for patients undergoing treatment. The goal would shift from a brutal war of attrition against the tumor to a more sustainable strategy of containment. This new way of thinking, sparked by the rediscovery of an old drug, could pave the way for a new class of cancer therapies that are not only effective, but also much gentler on the human body, offering new hope for patients and their families. It is crucial to remember that this research, while incredibly exciting, is still in its early stages. The experiments showing hydralazine's effect on glioblastoma cells were conducted in a laboratory setting, which is often referred to as preclinical research. These promising results will not immediately change how we treat patients in the clinic today. The next step involves a long and careful process of further investigation to determine if the same effects can be safely and effectively replicated in human patients. This will likely involve animal studies followed by meticulously designed clinical trials to test the drug's efficacy and safety specifically for brain cancer. One of the key challenges that researchers will need to address is the blood-brain barrier. This is a protective membrane that shields the brain from potentially harmful substances in the bloodstream. While it is essential for protecting our most vital organ, it also makes it very difficult for many drugs to reach their intended target within the brain. Future work will need to confirm whether hydralazine can cross this barrier in sufficient concentrations to have a therapeutic effect on a glioblastoma tumor. Researchers may also focus on developing new, more selective ADO inhibitors that are specifically designed to penetrate the brain and target tumor cells with greater precision. The scientific community has reacted with a blend of excitement and measured caution. The discovery is celebrated as a brilliant piece of scientific detective work that opens up a new front in the war on cancer. However, experts rightly emphasize that we are at the beginning of a long journey. The path from a laboratory breakthrough to a standard clinical treatment is filled with hurdles. More research is needed to understand the long-term effects of inducing senescence in tumors and to develop strategies to overcome potential drug resistance. This work will require time, funding, and collaboration among scientists and clinicians around the world. Even with the long road ahead, this discovery provides a powerful source of hope. The finding that a safe, inexpensive, and widely available drug could be repurposed to fight one of the deadliest cancers is a monumental development. It underscores the immense value of continued scientific inquiry and the potential for transformative discoveries to come from the most unexpected places. While we must be patient as the research progresses, this new understanding of hydralazine has opened a door that was previously locked. In the future, it could lead to better, more accessible treatments that help countless people affected by this devastating disease.